Hi, this is Mostafa Karimi. I will present Network Principle Deep Generative Model for Designing Drug Combinations as Graph Sets. This is a joint work between I and Arman Hassanzadeh under the supervision of Dr. Yang Shen. As we know, antibiotic drug resistance is a serious challenge for the whole world. Drug combination is a well-known approach to overcome drug resistance. It has been successful in many cases such as HIV, cancers, and TB. But the problem is that it takes decades to design a successful drug combination. The main question in the drug discovery is that how can we automate the process? The main part in the drug combination therapy is that we should target multiple components at the same time simultaneously in the same or parallel pathway. For example, if we only target MEK or BRAF alone in the melanoma, drug resistance will happen. But if we target both of them in melanoma, it will overcome the drug resistance. Drug discovery can be categorized into two groups. Generative models. Generative model will create completely new drugs. Or discriminative models. They will repurpose and reuse the current existing drugs. Current state-of-art or existing model in the drug discovery are single drug discriminative models. This model usually predict the property of a given drug. Single drug generative models. This model usually generates drugs based on a small representation or graph representation. But in the drug combination therapy, we only have drug combination discriminative model for up to K drugs. And they usually predict synergistic or antagonistic property of these two drugs together for a given disease. But there is no drug generative model for drug combination. However, there are challenges for creating and developing a generative model for drug combinations. For example, we need to drug combination, they should target multiple components in a disease, the genes or edges, but they should have low toxicity together. To overcome this problem, we have introduced a property called network biology principle. Another problem is that the search space will become bigger and bigger. Exponentially, it will grow with the number of the drugs. To overcome this problem, we have introduced a reinforcement learning model. As I said, we have developed the first model for drug combination generation. So our model consists of 4 sections. The first one is biological networks. The second one is HVGAE, how to embed or represent diseases. Once we find a good embedding or representation of diseases, we will give that to drug combination generative model or graph set generator, the RL model. This RL model will generate the drugs. Then we have created an oracle, which is based on a compound protein affinity prediction and a biological network to give us something called network-based reward, which I will introduce later. The compound protein affinity prediction is the model that we developed previously named Deep Affinity. Based on this network-based reward, the RL will learn to wisely and carefully design drug combination for a given disease. So formally, the problem of disease embedding is we want to embed diseases to points in RLD such that similar diseases are close to each other. But existing model only consider disease gene association and ignore a lot of information about it. For example, the hierarchy between gene, disease, and disease gene association, or other biological network information that we have, for example, cake pathways, geoterm, or heterogeneous gene relationship, the edge between two genes. For example, it can be regulatory, it can be signaling, so that there are all different information that we should utilize. Or, for example, each gene may not contribute equally to the disease embedding or disease representation. So we need to consider that genes may contribute differently to diseases. So main question is that how can we use all the information to do the disease embedding? In the next section, I will go over the HVGAE part, Hierarchical Variational Graph Auto-encoder, to do the disease embedding. Hierarchical Variational Graph Auto-encoder is a model we developed for disease embedding. For a given gene network, we use variational graph auto-encoder to embed them into a space of LG. Then, based on the disease gene association and attentional pooling, we will find out the contribution of each gene. And based on this contribution, we will find out and calculate the intermediate features for each disease. Once we know the intermediate feature for diseases, based on variational graph autoencoder, at the second level, we will embed or represent the diseases in the space of RLD. In this slide, I will briefly talk about the mathematics behind HVGAE. Simply, we have M type of features, geoterm, cake pathways, and we have NE type of edges between genes. For each type of edges, we have an agency. And based on this different type of edges and different features, we will learn intermediate representation of each nodes, or in here, each genes. We will embed or represent each node or gene as a Gaussian distribution. For Gaussian distribution, there is two parameters, mu and sigma. We will learn this mu and sigma based on the graph neural network of this intermediate feature we calculated based on these pH types and different features. Once we calculated the ZG, the stochastic representation of genes, we will use inner product as a measure of similarity between two genes. So if two genes are similar to each other, they will have high inner product, then there should be an edge between them. If not, there should not be an edge between them. Once we calculated the embedding or stochastic representation of the genes based on attentional pooling, we will find out the base or contribution of each gene. Based on the embedding and the contribution, we will calculate the intermediate feature for each disease. And again, through variational graph autoencoder and based on the assumption of Gaussian distribution for each diseases, we will try to represent them stochastically as ZD. And once we find out ZD, we can, again, based on inner product, we will find out the relationship between them. So if two diseases are similar to each other, there should be an edge between them. If not, there should not be. Finally, the objective function will be the reconstruction loss for genes, the reconstruction loss for diseases, and the KL distance between the prior and variational posterior in genes and diseases. And this loss function will be optimized based on Adam optimizer. In the next section, I will talk about the RL model that we have developed for drug combination generation. We have formulated the drug combination problem as a drug set generation. Our RL consists of K agents in which each agent will design a compound. The RL training process consists of three stages. At the first stage, we will make sure that each agent can design a proper compound. At the second stage, we will make sure that the agents can work properly together to design K compounds for a given disease. But we will do that across all diseases so that we can learn from different diseases. At the third stage, we will focus only on a specific disease and design for a specific disease. In this slide, I will talk about the state space and the policy network of our RL model. The state space is the intermediate generated graphs. The action space is which node or edge do we want to add to the current generated graph. For example, what is the first node that we should add? What is the second node that we should consider? What type of edge is between these two nodes? Is this the final step or not? So these are the four actions that we will do at every iteration. In this slide, I will talk about rewards. The first reward is chemical validity, to check whether the chemical bonds are valid. The second one is for the network biology principle. It is based on the complementary exposure, which I will talk about in the next slide. The third one is the generalized Slice-Wasserstein reward, to check whether in distribution the generated drugs are similar to the proper drugs, for example, the drugs in zinc library. I have talked a lot about network biology principle, but what is it? Suppose that we have a disease module and two drug targets. There can be 6 situations between them: complementary exposure, single exposure, independent actions, non-exposure, indirect exposure, or overlapping exposure. It has been shown based on the FDA-approved drug combination that the complementary exposure is the best one. And we have created a network-based reward based on this complementary exposure. So far, I have talked about our motivation, introduction, and method. From now on, I will talk about our results. We have compared our method with the state-of-art node embedding, the first set of deterministic node embedding, like node to wake and deep file. Second one is stochastic node embedding. These are only based on the disease. Then, if we consider both disease and gene level, but if we do not consider them as an end-to-end model, and if we do not consider the attentional pooling. Then, if we do train them jointly, but do not consider the attentional model, which consider the different weights for each genes for a given disease. And at the end, it is our own model. We can see that both the attentional pooling, joint end-to-end, and learning both gene and disease can contribute. And this has the more accurate results. Now that we have shown that our disease embedding is correct, then we have shown that actually our RL model can follow the network principle that we talked about. We have calculated the network principle based on the A XOR B, which is A minus B plus B minus A. And we have shown that for different disease categories from the disease ontology or disease ontology extended, we have similar or better network principle compared with FDA-approved network principles. Now that we have shown that our model follows the network principle, we will show that network principle actually correlates with low toxicity. We have compared our RL model with the 3 baseline models. At first, if you randomly select 2 drugs from drug bank, the gray one. Second one, if you select 2 drugs from the set of drugs available for a specific cancer, for example, for melanoma, we have 100 good drugs. If you randomly select 2 of them from the lung cancer, or if you select a drug pair from FDA-approved drug pair, for example, the drug pair that has been used for lung cancer, if we use it for melanoma or for others, can it be useful? So if we compare our generated drug combination with these 3 baseline, we can see that the red curve is actually better than all the other 3 baseline from the network score principle in all melanoma, lung cancer, ovarian cancer, and the breast cancer. And also, we have calculated toxicity based on a deep learning model called Deep DDI. This is an external deep learning model that we have used. We have shown that actually the toxicity related to our model is lower than all the other models. It shows that network principle correlates with low toxicity. Now that we have shown actually the network principle correlates with low toxicity, we will show in this slide that our design pair reproduce the pharmacology strategies. We have carefully chosen these 4 cancers actually, because melanoma cancer, it's related to same pathway, meaning that 2 genes that are important are in the same pathway. Lung and ovarian cancer, the 2 genes that are important are in parallel pathways. And for breast cancer, these 2 genes are not in parallel or same pathway. They have a very complex situation. And these are the FDA-approved drug combination that can overcome those resistance. Since we have shown that the network score matters, based on the network score and the intersection of the drug targets A and B, we have come up with a plan to just filter out the rest of drugs and come up with a specific drugs that we can further focus on. We have shown that actually for melanoma, 26% of the 69 drugs complementary and jointly attack the BRAF and MEK. And all of them actually attack the MAPK pathway, which these 2 belong to. For lung cancer and ovarian cancer and breast cancer, some of these genes we didn't have inside of our gene-gene network because of the gene-gene network is not complete. So we focus on the pathway level. And we have shown that actually out of this 896, 45% of them almost attack this MAPK and PI3K complementary and joint. It's a complementary exposure criteria. And 51% almost MAPK and PI3K for ovarian, and 9% of them for ER and cell cycle. And even, for example, for ER, for breast cancer, it has been shown that ER and PI3K pathway itself is important as well. So we have looked at the other potential pharmacology strategies for breast cancer, and 15% of them actually attack ER and PI3K in the complementary exposure way. In conclusion, we have developed two models, HVGA and RL-based graph set generator. In the HVGA, we have shown that it has an accurate embedding of disease network. For the RL model, we have introduced a network-based design principle in RL. We have shown that our model follows the network principle. It correlates with low toxicity, and we can reproduce the approved pharmacology strategies. Thank you very much for listening to my talk. If you have any questions, please email us. We will be more than happy to answer your emails. And our code, our drugs, everything is inside of our GitHub. If you have any questions, please let us know.